Good day. Good afternoon, students. Welcome to Raj Malhotra IAS Academy. Today in Science and Geography Current Affairs, we are going to discuss two important topics. One is very important. It's about rectal cancer, and I will talk about that rectal cancer in detail. In detail, I will cover that. Okay. Before that, one announcement for students who are appearing for HCS prelims on 10 July. We have a free Haryana test series, Haryana prelims test series. You will find it on portal. Okay, you can go. Description is given in link. Okay, description is given. Okay, now we start our topic. First topic is about rectal cancer, and second topic is about Ramsay Hunt Syndrome. Justin Bieber was in news about that. Okay, so first and most important topic is today's topic is 12 patients. 12 patients in USA have been cured for the rectal cancer. 12 patients in USA. This happened first time that a cancer cancer cells are completely destroyed, or you can say there was no sign of cancer after these clinical trials. Okay, and this is very important update, or you can say landmark, you can say research work in cancer therapy or cancer research. Okay, so we will discuss this in detail. First thing is, what is colon cancer or rectum cancer? Rectum, what is rectum? Rectum is part of your large intestine. Large, and the lowermost part of large intestine is your rectum. And the cancer of this, cancer means unlimited growth of this cells of this area, this rectum, unlimited growth of cells causes the rectum cancer. Okay, now what is the news about this? This cancer have been cured, completely cured by using a drug. That drug is actually a immunotherapy, or you can say is related to antibodies. And what are these antibodies, how they used it, I will talk in detail about that. The antibody they used is, wait a minute, antibody they use is dostrelimab. MAB is monoclonal antibody. MAB represents the monoclonal antibody. Dostrelimab drug, this is the name of the drug. Actually it is a monoclonal antibody. Okay, and we will discuss what are the monoclonal antibodies, what is the rectal cancer. Rectal cancer is the cancer of the cells or unlimited uncontrolled growth of the cells of rectum. And the rectum cancer are usually, listen carefully, rectum cancer are mismatch, listen carefully, this is very important, mismatch repair deficient cancer. This is one of the common type of rectal cancer. You will ask, what is this? Mismatch repair deficient cancer. Actually in our body, when cell division takes place, there is replication of DNA, and the replication of DNA is the process where mutation take place. How mutation take place? Due to mismatch of base pair, due to mismatch of base pair. For example, you have different base pairs, for example A, T, G, C, these kind of structure of DNA. Okay, so there are different kind of base pairs, and in mutation there is change in any base pair. For example, instead of C it is AT. Now what is the relation of this mutation and the cancer? Most of the cancer are due to genetic mutations, okay. In this case, in case of mismatch repair deficient mechanism, or you can say cancer. What happens due to this replication? After this replication, our cells have a repair mechanism. Repair mechanism is like by chance there is some addition, deletion, or some mismatch, the system, the cell system will repair the sequence, and what happens, the sequence, that repairing of sequence is deficient, means the system which is participating in that repair mechanism is deficient in these cancer cells, means these cancer cells have the ability to do more mutation. The cells have more mutation, and from these more mutation you will get the rectum cancer, cancer of the rectum. Now thing is, you should have an idea about mismatch repair deficient cancer. Means in these type of cancer there is defect in repair mechanism of cell during cell division. Means when one cell is producing two cells, two daughter cells, and two daughter cells, in third case they will produce four daughter cells. During that time, what is happening? During this time, there is replication of DNA, and replication of DNA, if there is deficit or there is some, you can say, defect in repair mechanism of DNA, then there will be more mutation. More mutation will cause the cancer or other diseases. Simple. Okay. Now in rectum cancer, this is very common. The most common type of rectum cancer is your mismatch repair deficient cancer. Fine. This is the type of cancer, and this is the most common type of rectum cancer. Fine. Now I will tell you about the monoclonal antibodies. Monoclonal antibodies. This is very important. What are these monoclonal antibodies? Can you see two type of these antibodies? First thing, you should have an idea about antigen and antibody. Antigen is nothing. Antigen can be protein, RNA, DNA, any molecule which is capable of generating immunity response in a body. Okay, which is capable of generating immunity response. I am coming, what is the function of dostarlimab? I am explaining that. Okay, so antigen is any chemical which can produce immune response in your body. Protein, it can be your carbohydrate or sugar molecules, anything which can produce immune response. Now immune response is produced by your antibodies or by your lymph cells, B lymph cells and T lymph cells, B lymphocytes and T lymphocyte. B lymphocytes are the cells which produces your antibodies, and antibodies can recognize any chemical specifically. For example, any protein which is foreign to your body, any protein, any carbohydrate, any bacterial cell, viral coat, anything it can recognize specifically. Fine. And these are antibodies. They will recognize it, generate immune response and neutralize it. It will neutralize it. Now there are two kind of antibodies produced artificially, or you can say by pharma industry. What are these? Polyclonal antibodies and monoclonal antibodies. Polyclonal antibodies and monoclonal antibodies. You should have an idea about a term called epitope. Okay, what is epitope? Antigen is a complete molecule. Antigen can be a big protein, can be a carbohydrate chain, anything. But epitope will be the specific area or specific part of that chemical which is binding with antibody. This is epitope. and the whole molecule is antigen. Specific region of your antigen which is binding to antibody is epitope. Now in polyclonal antibodies, you will have production of many antibodies which can bind to your antigen at different areas. Means there will be more than one type of antibody which will be binding to your antigen. Means it will recognize more than one epitope, These are polyclonal antibodies. They are very easily, they can be produced very easily, very cheap. I will discuss how they are produced. Monoclonal antibody will recognize only a specific portion of your antigen. Means they are very specific to a epitope only. They will not recognize another part of antigen. They are specific to a particular epitope. Now in this case we have used a monoclonal antibody. How they are produced, how polyclonal antibodies are produced, we will discuss that. Okay, simple procedure is there. Polyclonal antibodies are usually produced by injecting antigen into the body of especially a bigger mammal, from which we can take out large amount of blood, like goats, like your sheep, like your hare, rabbit. For example, in this case, the antigen is injected into the rabbit. Now, the immunity response will be produced in the body of rabbit. Simple. Immunity response will be produced in the body of your rabbit. Now, rabbit cells, B lymphocytes will produce antibodies. They will produce antibodies, and these antibodies are secreted in the blood of that rabbit. That blood will be collected. The blood will be collected, and from the blood, the antibodies will be filtered out, and they will be used for immunotherapy. They will be used. The issue is that the antibodies produced in such case are not from a single type of cell. They are from different. They are from similar cell but not identical cell. Means there can be some differences. There can be differences of epitopes. They will recognize same antigen, but at different, there can be mixture of different B type, or you can say different antibodies which recognize different epitopes on antigen. So this is the mixture. They are not very specific. They are not very specific to a single epitope, and this is the reason polyclonal antibodies have some side effect, because they can recognize other sites also. Fine. Now I will talk about monoclonal antibodies. What are these monoclonal antibodies? Monoclonal antibodies are produced from a single type of cells, single type of B cells. Now listen carefully. Rabbit or rat is immunization, or you can say antigen is put into the body of rat. It will produce immune response. And the spleen, the main organ which produces your B cells, B lymphocyte cell which produces antibodies. The spleen of the rat will be taken out. And the spleen will have B cells, B lymphocytes. These B cells will be fused with myeloma cells. Myeloma cells are cancer cell. Simple. Listen. Myeloma cells are cancer cell. This is hybridoma technology. Okay. The reason why we are using them, because these cells, B cell alone, are not reproduced on the culture in artificial medium. They cannot reproduce in culture in artificial medium. By making a hybrid cell of B cell and myeloma cell, we are generating a new type of hybrid cell which can produce from one generation, like cancer cell, they can produce unlimitedly, uncontrollable growth. And these B cells have genes for the B lymphocyte, and they will have genes for the antibody production. Single type, monotype of antibody will be produced. So these hybridoma technology will select the cells which can produce the antibodies which will recognize single type of your epitope. Only single type of epitope will be recognized by your these antibodies, and these antibodies are, you can say, then they can be produced by these cells and they can be filtered out, they can be collected and marketed. Getting my point? So in case of our this clinical trials, we have used dostrelimab, which is a monoclonal antibody which recognize a single type of, you can say, protein which will bind to a single type of protein. Fine. Now you have idea about cancer. Now you have idea about monoclonal antibodies. Actually, how this antibody, or you can say how this antibody works. Okay, listen carefully. This is the very important part. Actually, on cancer cells, you have a receptors, or you have a ligand PDL1, and you have a receptor PD1 on T cells. PD1 on T cells. When PD1 is recognized by PDL1, they bind with each other. They will bind with each other, and this combination does not allow T cell. It will not allow T cells to function properly. Means T cell will not kill the cancer cell till PD1 and PDL1 are together. When these two are together, the cancer cell will not die, T cell will not work or kill the cancer cell. Simple. Now, by using dostrelimab, or you can say monoclonal antibodies, what we are doing, we are blocking these two receptors. We are blocking these two receptors. Now, T cells and tumor cells cannot bind through PDL1 and PD1, and this allows the T cell to kill the tumor cells. It's very simple. There are two kind of receptors, or you can say there is a receptor and ligand which are binding from tumor cell and T cells. It makes the T cell inactive. T cells will not kill the cancer cells. What the scientists are doing, they are blocking the PD1. They are blocking the pathway of PD1. Now PD1 will not recognize the receptor PDL1. Now they will not bind, and when they will not bind, then T cell can function normally and it will kill the tumor cells. T cells, it will kill the tumor cells. Got it? This is very simple procedure. In this procedure, what is happening? We are using immunotherapy. There is no, you can say, radiotherapy or chemotherapy, means no utilization. Amrit, it's necessary because you should have an idea about monoclonal antibodies. You should have idea about what, and the thing is, in future these things will be regular in your current affairs, because this research is taking place in leaps and bounds. You should have basic idea about these things, and these are basic things. Simply, there are two receptors. One is ligand, they are binding, means the T cells will not kill. You should, this year there was question on T cells. This year there was question on T cells, was there or not? T cells are the immunocells which kills foreign cells or cancer cells. Okay. This year in prelims there was question on T cells or lymphocyte, you can say T cell and B cells. You should have an idea about immunity system. So cancer cells are killed by your T cells, and most of the cancer cell escape this killing by many method. In this case, the method is they are binding to the T cell and making this T cell inactive, and this cell will not kill the cancer cell. And by using, and this is the best way because it has no side effect. It has no side effect. Getting my point? No, in radiotherapy and chemotherapy, the patient's normal cells dies. You will find the hair loss, you will find the loss of other cells, and that is very painful for the cancer patient. This will not happen in case of this. Now issue is, this is in naive stage. This technology is in naive stage, means earlier stage, and other thing is this is very costly. This is very costly. This is around 4 lakh rupees per month, and the treatment is for 6 months. For a normal patient, the charges are very hefty. In future, maybe it become cheaper, but this is very costly. And other thing is, IPR related issue will come into existence after release of such kind of drugs. Fine, but this is a very big step towards the, or you can say in cancer research, very big step. Now another topic is you have seen, this was in news, okay, Ramsay Hunt Syndrome. सबको बताइए? हाँ जी, जस्टिन बीबर. His right side is paralyzed. His right side is paralyzed. Right face means he cannot blink this eye. He cannot smile with the right side of his lips because of Ramsay Hunt Syndrome. What is this syndrome? This syndrome is nothing. This syndrome is a viral infection. Is due to viral infection. Now you will say, how viral infection can cause this? Actually, viral infection is causing the inflammation of nerves. Inflammation of nerves. And this inflammation of nerves is causing the facial distortion, or the signal is not transmitted from brain to the facial nerves. This is nothing. The facial expression are movement of muscles. The signal comes from brain, and if due to viral infection, there is swelling, or you can say inflammation of your nerves, the signal will not be transmitted properly, and that is the main reason. It's not a permanent disorder. It is normal viral infection. And which kind of virus is causing this? Which kind of virus is causing this infection? Right, you should have an idea about this virus, because this is very Zoster virus. Zoster virus. This is the same virus which causes your chicken pox. Same virus causes your chicken pox. And the symptoms is, for example, you will have some rashes or some kind of blisters in your mouth, around your ear, hearing loss, and you will feel the weakness on the one side of your face. One side of your face. Fine. And this can happen normally. This is not a contagious disease. It will not like spread from one human to another human easily. Okay, ये spread नहीं होती है. Fine. And this is curable within 15 days to 3 months. The patient will recover. It's normal viral infection. It's not a genetic disorder. It's not a kind of, you can say, any due to damage of nerve or something. It's simply infection of a virus. That virus is causing inflammation of nerves, cranial nerves. What are cranial nerves? Which helps in movement of your face. Cranial nerves which help in senses, for example olfactory nerves, your nerves of your eyes, ears, which help you the sensing the signals of the signals, outside signals. In same way, you have cranial nerve 7, which is responsible for your facial. There are 12 cranial nerves in your body, 12, and cranial nerve number 7 is responsible for your facial movements, and swelling of that or inflammation of that will cause this paralysis. It can be left side, right side, doesn't matter, but it is due to the viral infection. Fine. Any issue with this? Fine. So I'm concluding this session. Again, you can join our HCS prelims test series. Prelims is on 10 July. These are very, very qualitative tests for Haryana prelims. It is not cumulable because of the site of infection. It's nerves. Nerves. Usually it is transmitted to the person whose immunity is very, very weak. There are many strains. There are many strains of these viruses. It's not the exact same virus which is causing chickenpox. This is different strain of same virus. Yes, yes. Other thing is vaccination is the other thing. And other thing is, in some cases, revert back of your vaccinated viruses. That can also cause these diseases. Fine. Chickenpox vaccination is usually normal. Chali. I'm concluding this. Thank you so much.